Good morning, students. Our today's topic is Chapter 9 Biomolecules. So, first, what are biomolecules? All the carbon compounds that we get from living tissues from our body can be called biomolecules. All the carbon compounds that are present in our body or in any living tissue are considered as biomolecules. So next is how to analyze chemical composition. So in the molecule, which fractions are present, which type of chemicals are present. For that, we can take any living tissue, a vegetable or a piece of liver, etc. From the chick's liver you can take or any vegetable and grind it in a trichloroacetate acid using a mortar and pestle. कोई भी लिविंग टिश्यू लिया, उसको ट्राइक्लोरोएसीटेट के साथ में मिक्स करके उसको अच्छे से मिक्स कर लिया विद द यूज़ ऑफ मोटर एंड पेसल. वी ऑब्टेन अ थिक स्लरी, एक मिल जाएगा, थिक सा मटेरियल मिल जाएगा. इफ़ वी वर टू स्टेन दिस थ्रू अ चीज़ क्लॉथ और कॉटन, वी वुड ऑब्टेन 2 फ्रैक्शंस. जब उसको हम स्टेन करेंगे, उसको एक फिल्टर करेंगे, एक पार्ट जो फिल्टर हो जाएगा उसको कहेंगे एसिड सॉल्युबल पूल, और जो ऊपर रह जाएगा रिटेंटेट वो होगा एसिड इनसॉल्युबल फ्रैक्शन. तो लिविंग टिश्यू को लेना है, ट्राइक्लोरोएसीटेट के साथ में उसको मिक्स बनाना है, स्लरी बनानी है, फिर स्टेन करना है. ऊपर जो रह जाएगा वो एसिड इनसॉल्युबल फ्रैक्शन होगा और जो नीचे आ जाएगा वो एसिड सॉल्युबल फ्रैक्शन होगा. नेक्स्ट इज अमाइनो एसिड्स. They are organic compounds containing an amino group and an acidic group as substitutes on the same carbon, that is the alpha carbon. Hence, they are called alpha amino acids. They are substituted methanes. There are 4 substituent groups occupying the 4 valence positions. These are hydrogen, carboxyl group, amino group, and a variable group designated as R group. This is shown in figure on page number 145 in figure 9.1, amino acids. The simplest amino acid is glycine. So in the amino acid, carbon is present. That carbon has the 4 valency. On one side amine, NH2, amine is present. One side acid, that is carboxyl group, COOH is present, carboxyl group. One is hydrogen, and on the 4th valency is balanced by the variable any R group. R can be H, CH3 or CH2, any group. R is the any methyl group. So the carbon has 4 valencies, one side amine, one side carboxylic acid, and one side R, and one is hydrogen. This is the structure of the amino acid. The chemical and physical properties of amino acids are essentially of amino group or carboxylic group and the R functional groups, because 3 groups are present: amine, carboxylic, and R functional group. So amino acid has the properties of all these 3 groups. Based on number of amino and carboxyl groups, there are acidic amino acids, example is glutamic acid, basic, example is lysine, and neutral amino acid, example is valine. At least learn one example of acidic amino acid, neutral amino acid, and basic amino acid. Similarly, there are aromatic amino acids, phenylalanine or tryptophan. In solutions of different pHs, the structure of amino acid changes. So here reaction is shown under different H-ion concentrations. It shows different types of structure. B structure is known as Zwitter ionic form. Zwitter ionic form. So what is Zwitter ionic form? Zwitter ion is a compound with no overall electrical charge, but which contains separate parts which are positively and negatively charged. In this B structure, positive charge is also present, negative charge is also present, but overall it is a neutral or no electrical charge is present on this. This type of structure is known as Zwitterion. If H ions are present, then A type of structure is formed. If OH ions are present, then C type of structure is formed. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl, ethyl, or higher number of CH2 group, that is up to carbon number 1 to 19 carbons can be present. Simple example is of palmitic acid, has 16 carbons including carboxyl group. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Fatty acids are found esterified with glycerol. Then they can be monoglycerides, diglycerides, and triglycerides. If one fatty acid is attached to the glycerol, it is known as monoglyceride. If two fatty acids are attached to glycerol, it is known as diglycerides. And if three fatty acids are attached, then it is known as triglycerides. These are also called fats and oils based on melting point. Oils have lower melting point and hence remains as oil in winter. Some lipids have phosphorus compound in them. These are known as phospholipids. They are found in cell membrane. The cell membrane is made up of phospholipids. Lecithin is the example of phospholipid. Living organisms have a number of carbon compounds in which heterocyclic ring can be found. In figure 9.1, some heterocyclic rings are shown below here. Some of these are nitrogenous bases. So in animals, 4 nitrogenous bases are present: adenine, guanine, cytosine, thymine in the DNA. In RNA, instead of thymine, uracil is present. So total 5 nitrogenous bases are present: adenine, guanine, cytosine, uracil, and thymine. Learn these 5 names. When found attached to the sugar, they are called nucleoside. When nitrogenous base is attached to the sugar molecule, it is known as nucleoside. And when phosphorus also attaches with the sugar and nitrogenous base, then this structure is known as nucleotide. So what is nucleoside? Sugar and nitrogenous base. When both are present, that structure is known as nucleoside. When sugar, nitrogenous base and phosphorus is present, then it is known as nucleotide. Adenosine, guanosine, thymidine, uridine and cytidine are nucleosides. Adenyllic acid, thymidyllic acid, guanyllic acid and uridyllic acid, cytidyllic acid are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. So DNA and RNA is the genetic material in humans. Next is primary and secondary metabolites. All the molecules which are present in animal tissue, they are known as metabolites, or we can say all biomolecules can be known as metabolites. That is primary metabolites because they are present in all organisms. However, there are certain other animals like in plant, fungus and microbial cells, certain other compounds are also present and they are known as secondary metabolites. Example is alkaloids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are the examples of secondary metabolites. In your book, table number 9.3, some examples of secondary metabolites are given. So learn at least one example of each. Primary metabolites perform some specific function, whereas we don't know about the function of secondary metabolites. Some are useful to the humans, like rubber, drugs, spices, scents, etc. And some secondary metabolites are of ecological importance to man also. Next, that is biomolecules, we will study next time. Thank you.